welcome to my YouTube channel. So today we are going to see some more applications of Biopython. So previously I told you in my previous lectures that if you want to concatenate two different sequences like DNA and protein sequences, okay, so there is a specific technique. So we will see how we can do that. So for starting from the basics, you have to install the biopython package by pip install python installation package, and then we will see how to write the code for concatenating two different sequences. Okay, so we will write from okay so we will write from bio.seq. We will import sequence. Okay, this module will import, and we will take one protein sequence. Okay, so we will take one protein sequence here. So we will write it as this one, EVRNAK. Okay, so this is our protein sequence. Remember that this is written in a one letter amino acid code format. So these are 6 different letters signifying 6 different amino acids. For example, V stands for valine, R stands for arginine, like that. Okay, and then we will take a DNA sequence also. Suppose it is ACGT. Okay, and we will concatenate these sequences. So we will write is equal to protein sequence plus DNA sequence, just basic addition. Okay, and we will print this. And then we'll run this cell. See, so this is printed. So first 6 letters are the 6 letters of the amino acid, and the last 4 letters are the 4 letters of the DNA. Okay, so if you write this as vice versa, suppose I wrote protein sequence first and then DNA sequence, so that's why in the concatenated sequence we get the protein sequence first and then the DNA. So instead, if we have written it as DNA sequence plus protein sequence, we would have got ACGT in the first place, then we have got the rest 6 letters. Okay, so this is a very basic technique of how you can concatenate two different sequences. Although concatenating two different sequences like protein sequence with the DNA sequence doesn't make much sense, but still if you want, that's why I showed you. So likewise you can concatenate any other sequence, like you have a nucleotide sequence with a DNA sequence also, like this. Now if you have a DNA sequence, or if you have any sequence, like you can change the case of the sequence. Okay, so let's see. So we will write again. I will just copy it. Won't write it again. So suppose you have we will again have to import this sequence module. So from bio.sequence import sequence, and we will write one DNA sequence, DNA underscore sequence. Okay, and this sequence is written as ACGT. Okay, now we want to get the uppercase letters of this sequence. Okay, so what we will do, we will just write DNA underscore sequence dot upper, and we'll run this cell. So the uppercase letter gets printed, ACGT. Okay, likewise if you have a lowercase, so suppose we will write it as, here you have the uppercase sequences written ACGT, and you want to like get them printed in the lowercase format, then you will just write dot lower, okay, and run this cell. So this gets printed in the lowercase, okay. So now we will see how to compare sequences. So again we will start with, say, this one only. We will just copy it. So we will have to import the sequence module. So suppose this is our sequence 1 and this is ACGT, okay, and we take another DNA sequence 2 which is the same sequence ACGT, okay, and we will take another DNA sequence which is slightly a different sequence. So let's see what happens. This is AGCT, okay. So third sequence I made a little bit different, and first two sequences I made them similar. So let's see what happens. So now I will comparing. So str string, because these are strings actually, sequence. So DNA underscore sequence 1 is equal to str DNA underscore sequence 2, okay. So let's see what we get the output. So it's true because both are similar, okay. So here if we do it the vice versa, so if we write it as str DNA underscore sequence 2, okay, is equal to str DNA underscore sequence 1. So this is, we are basically checking the two strings, okay, this first string DNA sequence 1 and second string DNA sequence 2. So we are checking whether they are equal. So when we are running, we are get the result as true, okay. So both are true, okay. So suppose then we check another one. So instead of comparing sequence 1 and sequence 2, we compare sequence 1 and sequence 3. So let's see what happens. So it's false. So this means that these two sequences are not similar. Similarly, we can compare sequence 2 and sequence 3 and run this cell. So this is also false, okay. So if you have a very large sequence of, say, a protein sequence or a DNA sequence, and you want to like same some 200 nucleotides, so you want to compare it nucleotide by nucleotide, okay. So maybe the nucleotides are of the same length, then only you can compare it with Python, and then you can get the result, okay, these sequences are similar or these sequences are not identical, okay. So in our subsequent lectures we will see more applications of Python in biology.